Another powerful technique used by researchers is that of separating molecules on the basis of their charge or on the basis of the pH at which their charge has an overall charge of 0. This phenomenon is called isoelectric focusing, and it involves a very interesting approach. To start with, one starts with a tube or a column of material that's full of charged molecules, some with many, many positive charges, some with many, many negative charges, and many molecules with small numbers of negative, small numbers of positive, and varying numbers of those in between. The interesting thing about that column is if I place that column in an electrical field, the molecules that are the most positively charged will go to one end, the ones that are the most negatively charged will go to the other end, and then I'll see a gradient of those all the way in between to the point where in the middle they'll have 0. Well, if I take a mixture of proteins and I place them in this mixture and I apply the electrical field, they will separate themselves in the same way. The most negative ones at one end, the most positive ones at the other end, and the most neutral or in charge in the middle. Well, if I do that, I've separated those guys on the basis of charge, a very, very powerful technique. But there's a more powerful use of this technique in the phenomenon called 2D gel electrophoresis, and so let me describe that to you. In 2D gel electrophoresis, one first does what I just described to you, which is to take this column of material, separate the proteins on the basis of their charge, and then take the result of that and then turn it, as you can see here, and place it on the top of a polyacrylamide gel. I add SDS to denature and make those rods that are negatively charged that I talked about, and then I take that material and I run gel electrophoresis separating in this way. Well, if I look across the top, I've separated from, let's say, the most positive on one side to the most negative on the other side. And now when I go down this way, I separate on the basis of size. So that when I get my final result, it looks something like what you see here, where I might have the most positive and largest here, the smallest and most positive here, the largest and most negative here, the smallest and most negative over here, or vice versa. All right? But in any event, I will have the smallest at the bottom, I'll have the largest at the top, and it depends on whether I have positive or negative according to which way I oriented the column. The beauty of this technique is I can separate on a single gel every single protein that's found in a cell, and I can see how much I have of it by the intensity of the spot that's on there. Now, this technique is really useful if I, for example, take a cancer cell and I do the technique, and then I compare the gel of the cancer cell to a normal cell, and I ask the question, which spots differ? Because then I will know which proteins are found in the cancer cell but not in the normal cell, or which ones are found in the normal cell but not in the cancer cell. So 2D gel electrophoresis, which is what I've shown you here, is an incredibly powerful way to compare protein production in different cells. The last technique I want to talk about here is related to that in a way, but instead of using proteins, it uses messenger RNAs. So this technique is called microarray analysis, and it's depicted here. It uses small glass slides that have been broken up, or not broken up, but actually divided into grids. And those grids are shown here by spots. So we might have a glass slide that has thousands and thousands and thousands of spots. And each spot has a sequence on it that's complementary to a specific gene. So I might have a sequence that's common, that's the complement to human growth hormone, for example. And on that spot where I have gene number 1, I would have thousands of copies complementary to gene number 1. Okay? Very, very important consideration. Gene number 2 would be a different gene, complementary to a different gene, and there would be thousands of copies there. So each spot has thousands of copies of the same gene, and each spot is unique to any given gene. All right. Now, let's imagine that I then take a batch of cells, let's say a cancer cell, And the cancer cell, I would take and I would bust open the cell and I would isolate all of the messenger RNA. So in this technique, I could take, for example, cancer cells, isolate the messenger RNAs from them, and place a dye on them that's blue colored. I could do the same experiment, take normal cells and isolate the messenger RNAs and place a dye on them that is a purplish or reddish kind of color, such as you see on the screen. I would then take and mix the messenger RNAs from both of these cells and then allow that mixture to hybridize with the microarray as you see here. Well, what's going to happen? If I allow the conditions to work properly, the complementary sequences are going to bind to each other. That is, the messenger RNA sequence, they're going to find their complement and bind, and they're going to leave a color behind in the process. So I wash away all the things that don't bind, and then I take a microscope and I look to see the microarray. What colors do I see? As a consequence of this analysis, what I can see is I might see something, for example, that's mostly blue. All right? Mostly blue would mean it's being made in the cancer cell, but not being made in the normal cell. Whereas something that's mostly red would be made mostly in the normal cell, but not in the blue, not in the cancer cell. And something that's a mixture of the two, let's say purple, might be present in equal amounts in both. So the advantage of a microarray is it's telling me not at the protein level how much is being made, but rather how much messenger RNA is being made in each of those cells. And the color, the intensity of that color is going to tell me how much and out of which cell that messenger RNA is being made.